If it's for fungi, okay, yeast or molds, it is in mitochondrion. Okay, you know that. So we don't do fungi here. So electron transport chain, what is the electron transport chain? Very simple example. Just give you a very typical one, which is NAD goes to FAD. NADH2 actually. FADH goes to FMM, goes to FES, goes to COQ, then we go to cytochroma C, then we go to cytochroma A, then we go cytochroma A3, and last one is oxygen. Okay, this is the electron transport chain. Let's say we give you a typical one. We mentioned about when we carry an electron, we transfer these electrons. We have energy is transferred. Now, how do we know this is a good donor? How do we know this is a good acceptor? Now, do you remember we mentioned about 2-half reaction? Because this is more positive, the charge, and this is more, this is more negative, and this is more positive. So when we based on 2-half reaction, we find any chemical which is very negative, that's potential to be an electron donor, and anyone which is more positive, we say it's an electron acceptor. And oxygen is very positive. That's why it is always at the bottom. Okay? Now you know all these things. Okay, we're going to mention that later on. Then, where is that catalyst? Or where is that oxidase, sorry? When the electron transfer from here all the way to the bottom, go to the cytochrome C, then go to the further cytochrome A, A3 or oxygen. In here, you need some help. That is called oxidase. So oxidase, if you look at the full name, it's a very, very long name. So you said it very easy, which is electron transfer from coenzyme Q to cytochrome C to cytochrome A. They need some of the enzyme to help. That is called oxidase. Now, electron transport chain is more obviously is in the bacteria need more energy to survive. Which bacteria needs? Especially they need oxygen as the final electron acceptor. Of course, it's aerobic bacteria. So if it's strict aerobic bacteria, usually it's oxidase test positive. Okay? So how are we going to do? Our test is very example. You get another neutron agar. The left side you're going to add is Pseudomonas aeruginosa. The right side you're going to add E. coli. Then after incubation, 35, 24 hours, we will add so-called a very long name. I already know the acronym, oxidase reagent. If you see all these turns purple color, which means oxidase test positive. Now, oxidase test you have to do in a very limited period of time is about 30 to 60 seconds. If you any time higher than that, longer than that, everything will turn purple because in the air there's oxygen. Okay? So that's a very tricky. You have to do it in 1 minute, within one minute. That's called oxidase test. Now, oxidase test in the clinical area is very important. Why? Pseudomonas aeruginosa and E. coli both are gram-negative. Both are log shape, and both grow dramatically. Can be we call residents in your intestinal area. So usually we're difficult to differentiate. So in the clinical area, we use oxidase reagent, oxidase test, can do a very quick identification. Now remember, which bacteria is oxidase test positive? When we talk about the examination 2, is Neisseria, is that right? Neisseria gonorrhea or Neisseria meningitis is oxidase test positive, and a very quick test for oxidase. Okay, so that's called oxidase test. Now we're going to move on to another fact sheet, which is called extracellular enzymatic activity of the bacteria. Now we mentioned about bacteria can use glucose, lactose, sucrose, all these kind of basic energy, which is a sugar, modified sugar. Now of course we know bacteria use glucose first, then they use lactose. We will mention this real quick in the examination 3 lecture. But you also need to know that bacteria can use other energy resource. For example, starch. For example, protein. And for example, we can use let me see here, all lipids. Okay, so we want to do a test. So that's why we have another called enzymatic test, which means how bacteria can use it. So bacteria can use lipids if they have lipase. Bacteria can use protein if bacteria has protease. And for milk, typically for milk, they have a casein, high amount of casein. So, which means if bacteria have caseinase. Number 3, if they have starch, bacteria also can use starch if bacteria has amylase. Okay? So we're going to do a test. How to do a test? It's very simple. We get 3 agar. What are those 3 agar? You can get it right here. The first agar looks like very heavy, milky. It's a milk agar. This is a milk agar. This milk agar, we want to see whether bacteria has protease, whether bacteria has caseinase. So we use skim milk. Why we use skim milk? Because no fat there. Do you know whole milk, how many fats there? 8 grams. 2% of milk, how many there? 1.6 or 2 grams. Skim milk is 0% fat. So it should be all is caseinase. Is casein. Okay? This is skim milk. Second one looks very bright. This is called sprint blue agar. This is the one we want to test whether bacteria has lipase. The last one, you're not going to see any difference. This is bacteria has starch. Whether bacteria can use amylase, has amylase can use starch. That's called starch. Okay, so very simple how we do. Split it to 2. Like we did it before. Of course, you're going to label. For skim milk agar, we want to see bacteria whether they have caseinase. On the left side, you're going to inoculate the bacillus. Right side, inoculate E. coli. For the sprint blue agar, we want to see whether bacteria has lipase. Left side, we inoculate Staphylococcus aureus. Right side, inoculate E. coli. For starch agar, we want to see whether bacteria has amylase. Left side, we inoculate the bacillus. And right side, we inoculate E. coli. Okay, very easy. Then we do incubation. If you see a transparent zone right here, which means amylase positive. If you see a transparent zone right here, which means lipase positive. Now the question is here, how do you know you have amylase? you may not see anything. There's something we need to add next week. Once it's grown, we need to add iodine there. Because starch will be reacting with iodine, turns brownish color. So, next week, if one side you see it's brownish, which means starch is still there, so no amylase. If you see no brown, which means it's amylase positive. So a little bit of twist on your mind, okay? If you see brown, no amylase. If you see no brown, that is amylase positive. Okay, so you have to add iodine there for starch. Okay, so today's work is very simple. Milk agar right there, spring brew agar, starch agar, you will do an inoculation. Can you turn that to the side? Then everybody come here. You need to grab. You have to record it. Yellow cap is sucrose. Green cap is glucose. The purple cap is lactose. And this is urea tube. Okay? So what you should do, urea tube is white cap. You need to have your personal rack come here to grab 3 tube fermentation urea test. Come here to get milk agar, sprint agar, starch agar. Don't forget, everybody get 2 nutrient agar. We will do catalyst test and oxidase test. Same thing, we split them onto 2. What gonna we do oxidase test? One side is Pseudomonas aeruginosa, right side is E. coli. How about the catalyst test? One side is Staphylococcus aureus, another side is E. coli. Okay? Now, all these plates, the median is gonna, the bacteria added to the median is fixed. And I will assign the bacteria what we're gonna add right here. Okay? These 4 tubes will be assigned a culture. These already been assigned. This guy, you guys gonna go do E. coli for these 4 tubes. You guys will be Staphylococcus aureus. You guys will be you guys gonna be Proteus vulgaris. Last group will do Pseudomonas. Okay? I said it one more time. 3 tube fermentation test and the urea test will be assigned a culture. So E. coli, Staphylococcus aureus, Pseudomonas, Proteus vulgaris, the last group is Pseudomonas. This will be already be assigned because it's not going to be changed. Where is the bacterial culture? PV, that is Proteus vulgaris. PA, Pseudomonas aeruginosa. BS, Bacillus. EC is E. coli. SA, Staphylococcus aureus. You get you wanted slants, please retain it back. Don't mess it up. This has to be retained back. And don't block it. Because I have another series, another 5 sections. Okay? So go ahead to do it.